right, this is finally we're getting to the last topic, theory of evolution, and unit 4.1, evolution of life. So, how did the first cell, the very, very first cell, come into existence? The theory is called spontaneous membrane formation. So, this is when, like a long time ago, long time ago, like 3.6 billion years ago, somewhere in the ocean, a few peptides, some of these, a few peptides, they lined up to form a membrane. They line up to form a membrane, and during this process, it somehow enclosed some RNA molecule with it. So some RNA molecule inside. And what happened is this RNA could replicate, and it acted as the genetic information for the early prokaryotic cells. Now, another role of RNA in these early prokaryotic cells is catalyzing reactions, somewhat like an enzyme. But in this case, it's not technically an enzyme because we know enzymes are proteins, right? They have specific 3D shapes that make them complementary to the substrate. But an RNA is not an enzyme because an RNA is not a protein. So we give it a new name. We call it the ribozyme. The ribozyme. All right. So ribozyme, like the enzyme, they can catalyze metabolic reactions, and RNA polymerase synthesizes RNA molecules such as tRNA, rRNA. And the fourth one, it could also catalyze the formation of proteins, because from the RNA, proteins form, right? Next, the evidence of prokaryotes coming first. There are four evidences. The first one is the DNA in eukaryotic cells are much more complicated. They have higher levels of organization than DNA in prokaryotes. So the first one is DNA. All right. Number two, compartmentalization. Not going to write that out. Eukaryotic cells have membrane-bound organelles in which different metabolic processes occur. So this is just saying, well, eukaryotes have more complicated cell organelles, which prokaryotes do not have at all. So I'm just gonna write down organelles, organelle. And the third one is very straightforward. This is just size. We know that eukaryotes are overall larger than prokaryotes. But we also have a fourth. I should put it here. And this is fossil record. This is the most straightforward one because the oldest prokaryotic cell to be dated is about 3.5 billion years old. That's pretty old. But the oldest eukaryotic cell to be dated is just 2.1 billion years old. So there's 1.4 billion years between the first, well, prokaryotic fossil and the earliest eukaryotic fossil. So that's just a lot of difference. So now we know where the first cell came from, what like how RNA functioned in the first cell, how they replicated, and the evidences for which came first, the prokaryotic or the eukaryotic. Now we know that. So we should know endosymbiosis. So this answers the question on how did eukaryotes come into existence, right? If prokaryotes just came from formation of peptides into a membrane, where did eukaryotes come from? Well, endosymbiosis is the word. Oof. Though, syn, yeah, biosis, endosymbiosis. We can break this word up into three parts. The first being endo. This just means into a cell. And syn, that's a very bad Y. Syn means living, and biosis, living together. We have, I think it's in year 11 biology, we have this word, and this just means two organisms living together. It can be a positive relationship. It can be a harmful relationship for one, such as a parasite, like a parasite on a horse or a pig. It's beneficial to the parasite because it's getting free food, free accommodation, but for the host animal, it's not a good feeling. But endosymbiosis is the process by which an early prokaryote, so a small prokaryote, an early old prokaryotic cell, enters a larger host cell. This is the process we refer to as endosymbiosis. So how it works is you have larger host cell, right? Larger host cell. And then you have a smaller early prokaryotic cell. It enters the host cell, but instead of being eaten up, digested, and killed, which is what usually happens, it's not. It continues to live inside the larger host cell and continues to respirate. Right? Over time, these two become interdependent. That means they become dependent on each other for survival. And if they can't survive without each other's company, that's a way to put it. So over time, this could become the mitochondria or chloroplast and make energy for the entire cell. There are four evidences for this theory. So the first one is independent replication. Mitochondria and chloroplasts are the two organelles that we theorize have formed from endosymbiosis. And this is because mitochondria and chloroplasts can both replicate independently of the host cell and through the process of binary fission. So I'm just going to write down replication. The thing is, we call this process binary fission, but in reality, binary fission, the word binary fission only applies to the replication of prokaryotic cells. We don't call the replication of organelles binary fission. We have another word for that, but the textbook just loves putting this word in here. So we're going to stay with that. And that's fine. Number two, double membrane. Again, mitochondria and chloroplasts, they have a double membrane. And they have it because they enter the cell, uh, they enter the cell through a process called endocytosis. Now, endocytosis is when a molecule enters a cell, right? Just a small molecule enters the cell. It doesn't just come in. When it does, when endocytosis happens, it gets another layer on the outside because it's actually put into a vacuum. And this vacuum is made up of the cell membrane. So the same thing happened to early chloroplasts and early mitochondria that came into the host cell. When they entered, they got the membrane of the original host cell around them, wrapping it up. That's how they got the double membrane for. Excuse me. And the third one is ribosome. So I'm just gonna write membrane. Okay. Mitochondria and chloroplasts have their own ribosomes that translate mRNA into proteins, so they can make their own protein And the ribosomes in mitochondria and chloroplasts have similar size and structure to those in prokaryotes. So RNA. And the fourth one is DNA. Like prokaryotic cells, mitochondria and chloroplasts, despite being organelles, have their own DNA. And this is single circular DNA molecules. Next thing, comparative genomics. This is the comparison of different organisms to investigate their evolutionary relationships. And there are four methods that scientists use to determine evolutionary relationships. The first one is amino acid sequencing. Amino a amino acid sequencing is basically just picking the similar protein from two different organisms and then comparing the actual amino acid sequence of the two proteins. The more similar they are, the more, well, generally, the more closely related the two organisms are. Vice versa, if they're not very similar, the two organisms are not distantly related. Second one is DNA sequencing. Now this is very straightforward. Same thing. If DNA are similar, the organisms are similar. If they're not, the organisms are not. So DNA. And the third one is also another type of sequencing. So this would be the rRNA gene. rRNA is ribosome. So the ribosomal gene between species. So the first three are all some sort of sequencing, amino acid, DNA, RNA. They all require you to already have the knowledge of the sequence of that particular organism you want to test. But the fourth one is called DNA hybridization, and this one is kind of special and different to the first three. Hybridization. Because this one doesn't require you to already have a knowledge of the DNA sequence or the RNA sequence or whatever sequence. What it does is it gets the DNA of from two different organisms you want to compare. So let's put one in black and one in red. It unwinds these two DNA. So they unwind, forming single strands. But then they cool the solution down to allow binding happen. So what happens is the um, the two single strands they together form a hybrid DNA. So this hybrid DNA is formed, but we don't just leave it there. What scientists do is heat up the hybrid DNA to a high temperature, and they record the temperature at which this hybrid DNA denatures and unwinds. And theoretically, the higher the unwinding temperature is, the more distantly related the two species are. And this is because if they are closely related, like a like like two species of birds, very closely related, they're going to have more similarities in their DNA. Thus, the two strands are going to be able to bind better with each other. Thus, they're not going to unwind as easily as the hybrid DNA of, say, two very different animals, like a cat or a dog. So that's how DNA hybridization works. And that's it. 4.1.